So now that we kind of had some time to take a peek at all the different components of blood, we're going to go ahead and dive into each of the types of formed elements, each of the type of blood cells we see. We're going to start with the erythrocytes. We're going to talk about their structure and function, and then look at the steps that are involved with the life cycle of an erythrocyte. So this is just a cartoon depiction of all the formed elements we could see in a blood sample. And we'll notice that the red blood cells are all these small red donut-shaped cells. The platelets are much smaller than everyone around them. And then our white blood cells, our lymphocytes, each of those types, because we see all 5 of the types we talked about in the last video here, all 5 of them look different from each other. So we see like a multi-lobed nucleus inside the neutrophil. We see a horseshoe-shaped one in the eosinophil. We see that H-shaped one in the basophil. The monocyte is very large, with a very large nucleus, and the lymphocyte is smaller without those granules. And so this is our shape of our red blood cells. Sometimes on occasion a cell may have a slightly different shape than this. That shouldn't happen very often though. There are some diseases that can cause changes in the shape of the erythrocytes, like sickle cell anemia, where they are shaped like a sickle or a crescent moon instead of this. There's also acanthocytes, which are red blood cells that have become almost like a starburst. But this is the normal shape of erythrocytes. They're kind of like a donut where the hole didn't quite go all the way through, so they look like a little pillow or like a little candy, right? Very smooth appearance, and we don't see any organelles, we don't see a nucleus inside of them. That's because those things actually were ejected out when the red blood cell was going through the maturation process. So these guys strictly are just for transporting things like oxygen around, and that's it. And speaking of transporting oxygen, that gets us to the major, super important component found on these red blood cells that allows us to do that: hemoglobin. Hemoglobin is made of 4 different chains, 4 different protein chains that have been bound together to form the entire protein, and each of these chains has what's called a heme group. The heme group here is that disc with the circle in the middle, and in that disc, right, that circle, is our iron. The iron is going to be what's going to help bind oxygen to the hemoglobin. So it's super important, right, that we have iron and that we can utilize iron to build this. Right? And this is why if you have low iron in your diet, you could have anemia, right, a form of anemia, because you're lacking the major, super important component to making this oxygen transport molecule. So now let's go ahead and look at the life cycle of an erythrocyte. Now keep in mind, if you remember what I said earlier, I said that these cells, these red blood cells, do not have all of the inner components. They don't keep their nucleus, they don't keep their organelles or anything like that. And so because of that, their life cycle is going to be relatively short. They're not going to reproduce on their own. We're only going to get more red blood cells using the stem cells we find in the bone marrow. So let's go ahead and follow from the bone marrow around to see what happens to a red blood cell during its life. So we start with hemopoiesis, which is the process, right, of making erythrocytes in the bone marrow, the red bone marrow. So we go into the red bone marrow, we have these stem cells that can lead to erythroblasts. Erythroblasts will be the precursor cells to the red blood cells, but we have an in-between stage of the reticulocytes. These reticulocytes then mature into erythrocytes, and they only live for about 120 days when they cycle through the bloodstream. So you think about it, that's only 4 months. So within a year, you've turned over all your red blood cells 3 times, right, on average. That's pretty crazy when you think about it. So these red blood cells, as they transport around, right, they may get damaged, they'll get old, and they'll need to be destroyed. So we have macrophages that one of their jobs is to find old and damaged red blood cells, phagocytize them, so basically bring them inside of the macrophage and then break them down. And we find these macrophages in various locations, including the bone marrow, the liver, and the spleen. So we will go ahead and break the red blood cells down, and then the protein portion of the hemoglobin, right, because there's a ton of hemoglobin on each red blood cell. I mean, we're talking tons and tons of hemoglobin molecules on every single red blood cell. So the protein part is going to be broken down into amino acids to get recycled. All the other components, right, the membrane, all that, we can recycle those as well. So we're going to use lysosomes to break down the red blood cells inside the macrophage. Remember, those lysosomes are kind of like those recycling factories. So all the amino acids, the cell components, those will go cycle through the blood. They can get back to the red bone marrow and they can be reused. But the heme groups, those are going to have a different fate, or at least they're going to have a slightly different fate. The heme portion is going to be broken down into biliverdin, right? So biliverdin, this is going to be for transport in the blood, right? The iron is going to bind to this protein called transferrin to transport around. We break down to biliverdin, and then anything that's unused, right, we can recycle back into hemopoiesis, or we convert it into bilirubin, which then we use to make BIOL. We can also transfer it to the protein ferritin, right? Fe is iron, right? We think the shorthand for iron is Fe, so ferritin will store it in the liver. So we have a couple things we can do with the heme group. We can use it to make bile, we can use it and store it in the liver, or we can just straight up recycle it to make more red blood cells. So we're really pretty efficient when you think about it when it comes to the erythrocytic life cycle. Now, like I mentioned before, we saw those nice, pretty pillow-like red blood cells that were nice and healthy, but not all red blood cells are necessarily healthy, depending on maybe a certain disease or certain genetic condition someone has, like sickle cell anemia. So in the case of sickle cell anemia, there is an error in how the hemoglobin is made and structured, and so that's going to change the shape of the red blood cells from that nice little pillow to a sickle or crescent moon shape. This is really dangerous to have. So if you have sickle cell anemia, you will need medical intervention, right? This is going to cause issues with the organs. It can cause issues with not being able to get oxygen around, clots, all sorts of things can happen if you have sickle cell anemia. So extremely dangerous to have sickle cell anemia, especially without medical care. All right, let's move on to our next one, which are the leukocytes and the platelets. So we get through this one, we're going to understand the general characteristics of leukocytes. We'll talk about their lineage and their main structural features and functions. We'll also talk about some malignancies involving leukocytes and then get into the platelets a little bit as well. So let's go ahead and talk about one of the cool things that white blood cells can do, and that is the fact that they can actually leave the bloodstream that the other blood cells are not capable of doing. So they can emigrate out of the bloodstream or go through what's called diapedesis. So the leukocytes in the blood, there's going to be some sort of chemical signal. We're not going to get into the details of this because that's more physiology, but there's a chemical signal sent out that is going to basically attract the white blood cells, and they're going to push their way through and squeeze between the cells that make up the capillary wall. So this is going to happen if we have some sort of injury, like a cut or a scrape, something like that, right? Something that has the potential to introduce pathogens into the body, right? And so we want to make sure that we destroy those pathogens to prevent infection. And the white blood cells are fantastic for that job. Now, the cut doesn't necessarily go to all these different blood vessels, and so we're actually going to not just wait to flow through the injury area. We're going to have white blood cells leave the bloodstream, push their way through the tissue, and find that cut and find those pathogens. They're following a chemical signal, right? It's basically like this, like a scent, right? There's a trail, a scent trail that they can follow. So the leukocytes squeeze between the capillary walls, which are very thin by nature, and they will move towards the injury and infection site. Once they get there, depending on the type of white blood cell, they'll have different jobs. Some of them will phagocytize the pathogens like macrophages or the neutrophils. Others will release chemicals to break them down, including like the neutrophils as well as eosinophils, right? So depending on the white blood cell that's there, they'll have different ways of destroying pathogens. Some of them, like I mentioned, the neutrophils, they can use chemicals or they can use phagocytosis. They're kind of multi-purpose. Other ones will only use chemicals, right, like an eosinophil or like the macrophages. These guys will just go in and engulf those pathogens. And so here we see some depictions of the white blood cells. We see the monocyte, which is a very large one with a very large nucleus without any grains. We see lymphocytes. In this case, we're seeing two of them because we have B and T lymphocytes. We will not get into the details of those because that is part of the immune system, but just understand there's multiple side types, I guess, or subtypes within that. The eosinophils we see with its horseshoe-shaped nucleus and lilac-colored grains. The basophil we see like those blue ones, and it's a much larger nucleus there. And then the neutrophil we see the grains, but we see that multi-lobes, right? In this case, we see 3 lobes making up the nucleus for the neutrophil. And then lastly, let's go ahead and get into the platelets. Now we're not going to get too deep into the platelets because again, a lot of what they have to do is going to be the physiology of clotting, but we do want to at least introduce them and know that these guys are actually smaller components of a larger cell that was broken up called a megakaryocyte, but these guys will be involved in clotting. We see how that in regular normal circumstances, we see like they're just kind of, they almost look like little flakes, like little oatmeal flakes, right? They're just kind of there in this picture, but then they get activated and now they look kind of like a little koosh ball, right? So why go through an activation process? If we think about it, it makes logical sense. You don't want your blood clotting all the time. You want your blood to only clot when it's necessary. And so having an activation system for something like a platelet is fantastic at preventing any sort of clots from developing that are unnecessary. Now in terms of how we actually clot, we're going to go real brief, real kind of over the top with this one. But in the bloodstream, let's say we have this injury, in this case a very square injury. You know, a piece of this blood vessel is broken open, erythrocytes are spilling out into the tissue or out onto the skin. That's not good. We have our platelets and we also have fibrinogen. Fibrinogen is going to then activate to fibrin, which will then activate the platelets. The platelets will then use different fibers like fibrin, like the activated form of fibrinogen, to form a net that will catch the erythrocytes. That is kind of sticky in a way that red blood cells will stick to. And so when you think of a scab, how it's red, right, and you're like, oh man, that's blood, right? You're not all too wrong, right? That scab is basically a sticky cell net that is made to catch red blood cells. So most of it is red blood cells with the platelets and the fibrin acting as that net to keep it all in.